from Sultan Qaboos University, Oman. Local chicken farming in Oman represents one of the main agricultural activities that provide opportunities for food security and income for many rural families. The majority of small and medium-scale farmers in Oman uses an open-sided housing system to grow their chickens. Greater understanding of the chicken gut function and microbiology can provide an opportunity for the improvement of chicken health and production in such environment. However, there is a limited information about the effect of house type on the composition and succession of intestinal microbial communities in birds raised in open-sided housing system. An experiment was conducted in the Poultry Research Unit at AES at Sultan Qaboos University with the objective to assess the relative abundance of bacterial microbiota identified in the intestinal tract of Co-500 broiler chickens raised in an open-sided house system from day 0 to day 35 of age using 16 srDNA-based analysis. Materials and method. One-day-old chicks Co-500 were obtained from a commercial hatchery and screened upon arrival to ensure that no abnormalities or symptoms of disease were present. Feeds and water were provided ad libitum, and 6 birds were randomly assigned to each of the 15 suspended wire cages, such that all cages had nearly a similar average initial weight. Birds per replicate combination were randomly allocated. To go over the experimental procedure, collection of gastrointestinal tract were conducted at 5, 15, 25, and 30 days of age. Sample of gastrointestinal tract contents were collected from 4 intestinal segments: duodenum, jejunum, ileum, and cecum. QIAGEN kit was used to extract DNA from the content of each lumen content samples. The DNA concentration was evaluated by measuring optical density using nanodrop 2000 at a wavelength of 260 and 280 nanometer. The integrity of the DNA extract was assessed visually using 1% agarose gel electrophoresis. The variable region Phi3, Phi4 of the 16 srDNA gene were amplified and sequenced. The PCR PCRs were performed in a triplicate. The PCR products were separated into 2% agarose gel and purified using the DNA gel extraction kit. Applicons produced from different intestinal luminal content samples and were sent to a commercial company, BGI Genomic Lab, China, for sequencing on the Illumina MI sequencing platform. The defined odds were assigned to different taxonomic levels, phylum, class, genus, and families at a cutoff of 97%. The clustered odds were also used to construct the refraction curves and calculate the Shannon-Simpson diversity indices, abundance-based coverage estimates, also Chaco-1 richness and coverage percentage by good methods. Metastats and R package were used to compare and determine which taxonomic group were significantly different between a group of samples based on intestinal segment and age period. Results. The alpha diversity indices were used to evaluate the microbial complexity in the duodenum, jejunum, ileum, and cecum. And when comparing the means of the indices, there was a substantial differences across intestinal region throughout the age period. This slide shows the percentage of relative abundance of bacterial community of CO500 broiler chicken determined from different intestinal segments at different age period from 16S rDNA libraries. Looking at the figures, species with an abundance of less than 0.5% across all samples were labeled unclassified. The intestinal segment, duodenum, jejunum, and ileum had a larger abundance of lactobacillus, and the percentage declined as the bird aged. However, in comparison, the cecum had a higher abundance of Clostridialis, and the percentage increased as the bird aged. This table shows the p-value distribution of 16 rDNA gene sequences libraries compared the abundance differences of microbial communities among samples from different segments for CO500 broiler chicken. The composition of the bacterial microbiota in the duodenum jejunum, duodenum ileum, cecum duodenum, cecum ileum, and cecum jejunum differed significantly, implying that each region established its own bacterial community. In the duodenum, jejunum, and ileum libraries, lactobacillus were the most common 16S rDNA sequences, while Clostridialis were the most common 16S rDNA sequences in the cecum libraries. This table shows the p-value distribution of 16S rDNA gene sequences libraries compared the abundance differences of microbial communities among samples from different segments for CO500 broiler chicken. The statistical analysis of the libraries revealed that the microbial composition at different age groups, day 5 to day 15, day 5 to day 25, day 5 to day 35, day 15 to day 25, day 15 to day 35, and day 25 to day 35 had no significant differences. In conclusion, our study shows that each region of different intestinal segment developed its own bacterial community, and the relative abundance was quite diverse. Future work should be directed to look towards histological alteration related to intestinal function, as well as should be focused by identifying gut bacteria that can be associated with improved or poor chicken growth performance. Would like to acknowledge the financial support by Sultan Qaboos University for this research project, and thank you.